It's really nice to be here today. Thank you, Bruce, for the invitation. So I'll be continuing the topic of chromatin, telling you a little bit about what we can learn from information contained within the histone methylome about the structure and function of the human genome and about the regulatory networks that drive both normal and malignant cells. So my talk has basically three sections. At the beginning, I'm going to talk about how we can use chromatin mapping technologies to learn a lot about the non-coding portions of the human genome. This is sort of the dark matter of the human genome, that beyond the genes, regions that have remained very poorly understood thus far, yet constitute roughly 95% of our genome. I'll then move on to talk a little bit about how we can use this systems approach to reconstruct the regulatory networks that appear to underlie cancer stem cell biology. And finally, I'll talk about some of the organizational principles of chromatin regulators, which may be relevant to those interested in, of course, targeting these proteins for therapeutic purposes. Okay, so I think you've heard a lot of background, but the genome is organized, wrapped around, DNA is wrapped around these nucleosomes composed of histones, and the histone tails are these hot beds of regulation subject to many different modifications, and it really depends on which residue in the tail is modified. You get different effects. There are enzymes in chromatin that either modify these tails or remodel chromatin or bind to these tails. So we've developed technologies to achieve systematic views or genome-wide views of the modifications on these histone tails across all the sequences in a human cell. This is a technology called ChIP-seq, and you've heard a lot about this already, but the idea is that living cells or tissues are fixed with formaldehyde. The chromatin is extracted and fragmented, and then antibodies against the modifications are used to enrich the portions of chromatin that have these particular modifications or have these particular flavors or states. Once that chromatin is pulled out, we deep sequence it with next generation sequencing instruments and basically count. We align the reads and then we count the number of reads at any given position, and this basically can be integrated into density maps across the genome that cover the entire genome and tell us about the peaks, tell you about sites in the genome where you have these particular modification states, and you saw some of that from Jonathan and from Scott, so I won't belabor this too much. In a large project under the ENCODE consortium, we took a whole panel of about 10 different cell types and about 10 different modifications, and you can see some of these are activating modifications, but also some of these we've also looked at some of the repressive modifications, right? It depends very much on which histone residue you're modifying and which tail. We've mapped these comprehensively. Altogether, we had about 200 different genome-wide ChIP-seq experiments, and we teamed up with some colleagues at MIT, Manolis Kellis and Jason Ernst, some computer scientists actually, to try to make sense of this vast amount of data. I'm not going to go into all the details here, but basically by looking at the different combinations of modifications that can exist in the genome, one learns about different types of chromatin states that can occur in the genome, and really there are about 100 modifications, and we're looking at about 10 of them here. There is a huge number of sort of conceivable patterns one could see, but when you actually look at the data, there are a relatively limited number of states that the tails occur in, or a limited number of combinations. So we were able to use Markov models to derive these different combinations. And we basically derived about 15 different states. Each of these states is associated with different kinds of modifications. Like here are some active states associated with lysine 4 trimethylation and some acetylation marks, whereas there's other examples of repressive states, or these are actually transcriptional elongation states, as well as repressive states associated with repressive modifications. So using this information and going back to the genome, it quickly becomes apparent that you can use combinatorial histone modifications to annotate the genome based on these patterns. The first set of states really correspond to promoters that are either active or weak, or promoters that are poised, such as the type we saw in the last talk. Other chromatin states or patterns help you identify enhancers, which have been particularly difficult to identify in the genome as they are not sites of transcription. They are not necessarily associated with exons and indeed can be very far away from the genes. One can also identify sites that function as insulators to block enhancer functions, and so on. So as a consequence, we can basically take this data and we can come up with a single genome-wide annotation per cell type that tells us where all the promoters are, where the transcripts are, and where the enhancers are, and also tells you about which enhancers are either active or poised in that given cell type. So I'll just give you a very brief sort of overview of what kind of things we can do with these data. Let's see. We can cluster enhancers. We can identify the enhancers across the genome. We found about 100,000 different enhancers across the genome. These are these short sequences that act at a distance to regulate gene expression. We can validate them in reporter assays in tissue culture models. We can cluster them based on their activity in different cell types and identify their cell type specificity, and then use this information to then link them to putative target genes that have appropriate types of functional activity. So for example, enhancers in immune cells tend to be near or linked to target genes that have functions in immune response, and so on. And we can also predict the transcription factors based on the enhancers and the motifs that the enhancers contain. So for example, in embryonic stem cells, enhancers that are quite specific to embryonic stem cells tend to have motifs bound by OCT4, as you might expect, and this is probably underlying their very specific activation in embryonic stem cells. Okay, so what can you do with this kind of genome-wide annotations now of the different types of sequence elements in the genome? Well, we wanted to apply this to some human genetics, and specifically to the field of genome-wide association studies, where many studies are undergone right now to identify the sequence variants, the differences between individuals that are associated with disease. So there's some rate of every 1,000 base pairs or so differ between individuals, and there are now studies, a number of studies ongoing to identify that subset of variants that are associated with specific diseases. There have been probably now more than 1,000 different genome-wide association studies for nearly 200 different phenotypes. And through these studies, thousands of single nucleotide polymorphism have been identified, or variants that are associated with particular phenotypes or disease. As has been widely reported in the literature and in the press as well, one of the sort of surprising results of the genome-wide association studies was that a vast majority, actually something like 90-something% of the sequence variants associated in these studies were non-coding. So these sequence variants aren't affecting the proteins. They're not changing the amino acid sequence of the proteins. Rather, they're acting in promoters or introns, or most often in intergenic regions sort of outside of the genes, really suggesting that they're probably playing some kind of regulatory role to titrate gene expression. So we took a look at these data sets. We crossed these genome-wide association data sets, databases of sequence variants, against our database now of annotated elements, including enhancers and insulators and so on. And what we were really struck by is that the greatest enrichment for the non-coding variants was in the enhancer elements themselves. So these sequence variants tend to coincide with our annotated enhancers. And moreover, there was a correspondence between what kind of study those variants were turned up with, what's the disease association, and what's the cell type in which the enhancer is active. So here's just a number of examples. So the top association we pulled out in a totally unbiased approach was a genome-wide association study for erythrocyte phenotypes. And the variants, the sequence variants, are SNPs between individuals that are most associated with different erythrocyte phenotypes, are enriched in enhancers that are specifically active in K562 cells, which is an erythroleukemia cell line. If you then look at the second hit was studies of lipid levels identifying those kind of variants, and those are enriched, as you can maybe guess, in HepG2 cells, which is a hepatomatous cell line. And then there are a number of examples, for example, then of immune-related diseases where variants associated with those immune diseases are frequently sitting in enhancer elements that are active in different lymphoid or immune cell types. So I'll just give you a specific example of that. This is just showing the actual data for the erythrocyte phenotypes. These are the different sequence variants as annotated as such. And we can basically, these pictures here are telling us, the colors are telling us which of these variants are annotated in our chromatin state maps and how they're annotated. And I think you can see that here's a large collection of variants that are annotated in orange or yellow, which reflects that they have enhancer state or enhancer activity in erythroleukemia cells. We can go on and also take some of this information on the sort of putative gene targets that are correlated with the enhancers and thus help not only identify to triage which SNPs should be followed up from these studies, but also predict gene targets. And finally, we can look at these variants themselves directly, and we can ask, do they affect any of the transcription factors that we are predicting as being upstream of the enhancer elements? And indeed, there are a number of cases where we can now begin predicting how these variants might be changing a motif that could then affect the binding of a transcription factor. So a database, a number of associations between the chromatin state maps and human genetics, and really an opportunity now to predict functions of these disease-associated loci, to predict the causal mutations in relevant tissues. So I hope this has just given you a little bit of insight into how these chromatin state maps can identify regulatory elements and the regulatory state of the genome. I wanted to go through this pretty quickly because I wanted to move on to some of the cancer studies. But maybe seeing some of this data and how it's connecting with human genetics will spark your imagination now about how we can apply some of these same techniques to understand something about the regulatory state of cancer cells, in particular of cancer stem cells. So together with Miguel Rivera and Daniel Haber, we sort of launched a pilot studies a couple years ago, a study of Wilms tumor. So Wilms tumor is the most common pediatric kidney cancer. It presents before age 5, so it presents very early, which is, of course, really critical to this disease and to the study. Like many pediatric tumors, it carries very few genetic aberrations. There are mutations in WTX and WT1, as Daniel has shown in his laboratory, but many of the other mutations associated with adult cancers are extremely rare. So P53 mutation is extremely rare in these tumors and was not present in any of the tumors that we're studying here. Another interesting aspect of Wilms tumor, in addition to these very few mutations, it's been causally linked to an epigenetic event, that is, to the loss of imprinting of IGF-2, which is normally only expressed from the paternal allele, the chromosome inherited from the father, but in certain conditions is expressed biallelically to higher levels, and in those cases there is a causal association to Wilms tumor. So this was an attractive tumor, both from the concept that this is genetically pretty normal, so there must be an epigenetic event that suggests the importance of epigenetic events, but also from a very practical perspective. The tumors are resected and large, and there are these pretty substantial fields of these very blue blastemal cells that look a lot like embryonic kidney, and which can be then resected and basically leave us with a relatively homogeneous field of cells that we could then do the chromatin state analysis in. So let me just, we took this data, and Aviva Presser, a grad student at the Broad, really looked at it in a pretty unbiased fashion. A number of us in this field had noticed that while most genes have a very sharp peak of lysine 4, this activating mark right over their promoter, there are, in different cell types, a fraction of genes on the order of dozens or maybe 100 genes that have something very different. There are very broad domains of lysine 4-ME3, and so for an example of this would be the Hox loci that Scott was talking about earlier, very broad domains of this activating mark, very distinct from what you see at most genes. And there's a tendency for these broad domains to sit over developmental regulators, such as HOX, but also a lot of other genes like that. If one takes a completely unbiased approach in embryonic stem cells and asks for the largest domains of open chromatin in a cell, you'll pull out things like OCT4 and SOX2 and NANOG. You'll basically pull out the pluripotency network that drives embryonic stem cells, even if you didn't have any a priori knowledge of the stem cells. So if you do this in Wilms tumor, which we did, you'll pull out, actually very interesting, you'll pull out IGF-2, which is causally associated with Wilms tumor, as I told you, but you'll also pull out genes like 6-2, DOC1, and GDNF. And if you then look these up in the literature, you see that these genes, which are associated with very active chromatin in Wilms tumor, which are highly expressed in Wilms tumor, really correspond to a well-described network of kidney development. So these are the genes that we know from studies in mouse development these are essential for developing a kidney. They fit together in these nice networks. Really suggests that Wilms tumor is, from a transcriptional regulatory perspective, mimicking a normal renal progenitor. So it looks like a normal renal stem cell in terms of its network, its transcriptional network. Let me give you a little more information too, though. If you then look at some of the epigenetic regulators, of the factors, the chromatin regulators that control the epigenetic state of the cells, here you can see Wilms tumor. Here's a number of genes that are highly expressed in Wilms tumor. You can see that it doesn't look much like an adult renal cell carcinoma. These genes are not highly expressed in renal cell carcinoma. They're not highly expressed in adult kidney, but you see this red stripe here suggesting these are highly expressed in fetal kidney. So in terms of its epigenetic state and chromatin regulators, again, the Wilms tumor is looking a lot like a fetal kidney. And finally, I'll just point out that P16, which becomes methylated at the DNA level in so many adult tumors, is hypomethylated, does not have DNA methylation in Wilms, but rather is associated with polycomb repression in this pediatric tumor. And this is a very characteristic landscape seen when one looks at normal, non-malignant tissues. So in many ways, then, Wilms seems to be mimicking a normal, what we think of as a normal renal progenitor that has somehow arrested its differentiation. So yeah, let me just build upon that here. The idea is that the hypothesis is that the Wilms cell is really mimicking this normal renal progenitor in terms of its epigenetic state and its transcriptional regulatory network. There are not so many changes needed to lead to arrested differentiation and thus transformation in this context. And I guess I would sort of hypothesize and contrast this with an adult renal cell carcinoma, for example, where these renal progenitors are no longer present. They sort of peter out by about age 5 in a kid, so you need to take presumably more specialized kidney cells and then reprogram them to cause renal cell carcinoma. And of course, renal cell carcinoma was one of the first of now many, many tumors that was found to have many genetic mutations in the chromatin regulators, which we would speculate might be required for some kind of epigenetic reprogramming to achieve sort of an immortal state here. Okay, so I want to shift gears now and talk a little bit about some studies in the lab by a number of my colleagues. Actually, a collaborative study with Hiro Wakimoto, Esther Reinbay, Andrew Chi, and Mario Suva in pathology, where we're now looking at adult glioma. So I told you that a big advantage of the Wilms tumor is we had a clear field of these blastemal cells that we could study, homogeneous population. This is clearly not the case in brain tumors. So we went straight away to one of the relatively now well-established models for tumor stem cells, specifically a brain tumor stem cell model, glioblastoma cancer stem cells shown here, where you have neurospheres that can be grown in serum-free conditions in the presence of growth factors. They express CD133 and other markers associated with stem cells. And Hiro and Samuel have clearly shown that these particular patient-derived stem cells will recapitulate tumors in mouse models. So Andrew and Esther began this project by taking these GBM stem cells, growing them in the lab, subjecting them to the same ChIP-seq analysis that I showed you before to identify the regulatory elements and characterize their state, right? And of course, they needed some comparisons, so they used neural stem cells and astrocytes as normal. And their goal here was to identify, like I showed you for Wilms now, a transcriptional regulatory network for the glioma stem cells. So I'll just show you a little bit of the data to start with. Here's sort of a comparison between at the top showing some normal stem cells, normal astrocytes, and then here are the next four are the various GBM stem cell tumors. The green is one of the marks associated with transcriptional activity, and the red is a mark associated with repression. Here are just three sort of well-known transcriptional regulators, ASCL1, HEY2, and KX2-2, that are highly active in virtually all of the GBM stem cell models from different individuals that we're studying, but that are inactive in the normal neural stem cells and the normal astrocytes. So this is just looking at the genes and identifying which transcription factors are active and which are inactive. But remember now with the chromatin maps, we also have information about the enhancers, sort of the distal regulators that are then integrating these signals and forming networks to drive expression. And here you can see in the presence of ASCL1, there is not a lot of action going on outside of the gene in these normal cells, but that in the GBMs one can find little of these light greens, and also in the presence of OLIG2 and OLIG1, same thing. These are really characteristic features of enhancers. So we're now beginning to pick up enhancer elements that are specific to the tumors. And then we're able to pull out the motifs. And Esther Reinbe is using this to begin to integrate early, but beginning to integrate the transcriptional regulatory networks that are sort of, we think, are driving the state of these cells. The data are fairly preliminary. We have about 25 transcription factors that we think their high expression, sit in a module, drive the brain tumor stem cells. ASCL1 seems to be sort of at the top of the chain here. If you knock down ASCL1, the cells lose their clonogenicity. We also see that the module seems to be upstream of the Wnt pathway, which we heard about earlier before. Again, if you knock down ASCL1, you lose the Wnt signaling in this model. And I won't go into the details, but the different parts of the module really correspond and appear to reflect some of the different types of GBM subtypes that one sees in the clinic. So we wanted to go a little further with this and see if we could learn something about the epigenetic heterogeneity of these tumor models. So I told you about the GBM stem cells shown here. These are the neurospheres. But I didn't tell you is that if you take the same tumor from the same individual and you put it under serum conditions, you end up with these more adherent spindle-shaped cells. These cells cause tumors in the mice. These cells don't. The stem cell model has very high expression of CD133. The serum model has almost no expression of CD133. So Maria Suvo in the lab wondered if it would be possible to take this transcriptional regulatory network, this module of transcription factors, and use this information to drive cells from here to here, right? So if you take these FCS cells and you plate them under serum-free conditions with the growth factors, you get virtually nothing. And the question is, is there a way to overcome that? So here's just some pictures showing you take the sort of these non-stem-like cells, you put them into the stem-like conditions. The cells do not form neurospheres, they all die. You don't get expression of CD133. And then Mario's sort of taken the Yamanaka-esque approach of taking the transcription factors from this regulatory network that were derived from the chromatin maps, putting them in successively, 15 TFs down to 3 TFs, getting a little progress. And most recently, he is identifying certain combinations of just a few different TFs that that will take these differentiated GBM cell model and appears to turn it into a stem cell-like model, gives it the capability to grow in neurospheres, and we're currently pursuing these in animal models to see if they really are cancer-initiating cells. Okay, so just for the last couple minutes, the last short section of my talk, I want to come back to the chromatin regulators that I think we've heard a lot about in several of the other talks. These are, the histone modifications have been sort of extensively explored by these mapping technologies. We really have used them extensively to annotate the genome, understand where they sit. But very few of the chromatin regulators, these enzymes that put on the marks or remove the marks or bind the marks, have been localized, and this is due to technical issues. The antibodies are lousy, the proteins are sitting there a little more peripherally to the DNA, and they're just very hard to study. This is a real problem for the chromatin aficionados because you need to have something to knock down, perturb by RNAi to understand the function at the locus, and there's no way to knock down a histone modification, right? Or unless you can understand the chromatin regulator that's targeting it. So Oren Ram and Alon Goran undertook sort of a heroic screen in the lab where they somehow coaxed out of antibody companies 150 or so antibodies to different chromatin regulators. They developed an assay called ChipString that uses this nanostring device to very cheaply and rapidly screen antibodies and conditions to try to find immunoprecipitation conditions where you can actually selectively pull up parts of the genome. They did this. Most of the antibodies fail, don't enrich anything specifically, but they identified about 29 antibodies to different chromatin regulators that did seem to enrich chromatin specifically. They then applied these to our ChIP-seq pipeline, and sure enough, they were able to map these 29 chromatin regulators in K562 and some subset of those in ES cells as well. Let me tell you a little bit about what they found. So this is just a list of some of the regulators. You'll recognize many of your histone deacetylases, some of the proteins that bind to the methyl groups, polycomb regulators, P300, and so on. What was striking was that virtually every single one of the regulators that we studied co-bound with other regulators in particular combinations. So this map here is just telling you about the correlations in binding between two regulators. So the diagonal is purple all the way because something correlates with itself at 100%. But what you see of these off-diagonal squares is telling you that all of these regulators shown here in group 1 are kind of binding to the same places in the genome. Same for these regulators shown here and these regulators shown here, and to a lesser extent these guys shown here. So we were able to use this information to now impute the modules. So really come up with a model that these regulators are not acting all by themselves, they're acting in modules. Some of those modules reflect chromatin complex or complexes of proteins that are physically tied together, but other modules really reflect just common modes of binding to targets. We can look at where these modules are going. So this is the first module, and this is a bunch of regulators, including both positive regulators and repressive things that you characteristically think of as having repressive activities like HDACs. These guys really sit at the green sites in the loci, which are active and competent promoters. So you've got these guys sitting at TSSs. These guys are all sitting at sort of enhancer elements, and you might have heard of P300, which has been discussed extensively as a regulator of enhancer. LSD1 is a demethylase that sort of probably is helping to keep that homeostasis of the K4ME2 because LSD1 actually removes the marks associated with the enhancers. And then this is another module that has kind of got diverse associations, but in many cases binding to enhancer elements. So the modular structure and organization of binding, we wanted to do a little more careful analysis. So now we took a very precise sort of fine scale binding analysis of the promoters. Each one of the rows here, it's hard for me, the pointer is dying here, so it's hard for me to point, but each one of the very fine rows corresponds to a different promoter in the genome, and there's about 1,100 of them shown here. And I think you can see that the different regulators sort of correspond to different sets of promoters, all the way up here from the upper left-hand corner, where you have a number of regulators that bind very active genes, all the way down here to the lower right-hand corner where you have chromatin regulators associated with repressed genes. We clustered these promoters and their genes according to the patterns of chromatin regulator binding. And two things really jumped out at us. The first thing is that generally the binding corresponds to the modules I showed you in the last picture. But there's also occasional swapping of modules, suggesting that not only is there a modular organization, but there's also a combinatorial association. And this looks a lot like transcription factors. Okay? The second thing is that there's specificity in the gene targets. You might think that chromatin is a structural entity. It's driven by the transcription factors and where it goes, but I don't think that's quite true. I think that there's this kind of data showing that there are specific subsets of regulators that go into cell cycle genes, to chromatin architecture genes, or to protein metabolism genes tells you that there's a degree of specificity in the targeting of chromatin regulators that I think is somewhat unanticipated, and that the structure and organization of this network looks a lot like transcription factors. So just to conclude, then, we now have, I think for the first time, pretty detailed maps of the different kinds of regulators that are sitting at enhancers, sitting at active promoters, sitting in genes, sitting at repressed loci in differentiated cells or in ES cells. There's a modular and combinatorial organization. The modules have opposing activity. This is all a chemistry experiment. You have acetylases and deacetylases sitting at the same places and sort of battling each other to reach homeostasis. And finally, the target gene sets imply some high specificity. And in particular, starting to pick up that a lot of the genes that are mutated in cancer tend to be associated with some of the chromatin regulators that are mutated in cancer tend to be associated with a lot of the enhancer elements or associated with the differentiation-associated genes. Very speculative, but it makes one wonder if these hits that one sees in the sequencing studies of the cancer reflect some fitness events to affect chromatin regulators that might somehow dampen down differentiation to some degree. So let me just conclude the talk then. I've told you how epigenomic or chromatin state mapping can tell us about the structure and regulation of the genome, how we can identify elements, and these functional annotations facilitate interpretation of sequence variants from GWAS. I've told you about how these same technologies can be applied to cancer cells, in particular cancer stem cells. In the case of pediatric Wilms tumor, it looks like there's a regulatory network that really mimics normal progenitors that have been arrested, whereas adult GBM seems to reflect more cellular heterogeneity. There's a regulatory network that doesn't really look like anything in normal physiology, but seems to be quite specific to these malignant stem cells. And finally, I've told you about the modular and combinatorial organization of chromatin regulators and some unanticipated specificity in their targets that could explain something about the mutations we're seeing in the cancer sequencing studies. And I think I've mentioned my colleagues along the way, Esther Reinbay and Mario Suva, in particular on the glioma study. I hope I mentioned Aviv Regev and Noam Shoresh, who collaborated with us on the chromatin regulator work, and of course Manolis Kellis at MIT. Thank you very much.
Hey, Brad, I have a question for you. So you have in cases where H3K4 tri is very sharp at transcription start sites, and then you had those broad domains for the developmental genes. Do you think that is causing something different to happen? I mean, what would be the rationale or reason? Because it doesn't look like it's based on gene size.
That's a great question. I think it's epigenetic. I mean, it's the master regulators have great big CPG islands, and they have great big regions of K4 domains, or under their silence condition, they have great big regions of repressive domains. If you think about it from the perspective of epigenetic inheritance, when cells divide, the histones segregate randomly. So if you want to remember the histone pattern, or if you want to have an epigenetic memory of a histone modification, you better modify a big region. So there's a lot of histones that are modified. So when you segregate them during division, you still have enough of those modifications to propagate the state. So I think it's epigenetic. I think that's why you're seeing this in the Hox loci. I think that's why a lot of the master regulators have these patterns.
And so, along those lines, do you find the other modifications look similar? Meaning, is it just H3K4 trimethyl that's spread like that, or other things spread? You said K27 probably.
So K27 under the repressed state spreads. K36 does not have the same patterns. We haven't looked at K79ME2, but it looks like that's a very good candidate for having. I don't know, what would K79 look like?
We probably can look. We have the data.
One should look.
Yeah, haven't looked at it.
Yeah.
Another question for you. In your GBM cancer stem cell modules that you're developing, you're doing H3K4 trimethyl profiling, K27 trimethyl, and K4 monomethyl.
Yeah.
Are those, so in our hands anyway, K4 trimethyl is not very good at predicting whether or not a gene's expressed, presumably because it's poised and not fully. Yeah. Does the monomethyl help?
The combination of K4ME3 plus K27ME3 is at developmental loci is pretty good at predicting. You're absolutely right that if you only had the K4ME3 data, you might need more. But we use the combination of K4ME3, K27ME3, and I didn't show it here, but we also use K36ME3.
Yeah, so that would help a lot, yeah.
The K4ME1 is different. The K4ME1 tells you about the distal elements. If you think about it, transcription factor is expressed, and then its binding places, and probably what it's doing is not binding to promoters. Increasing evidence suggests it's binding to distal regions, and that's what's driving the cell type specific expression in cells, right? So the distal elements are telling us about the network.
So,
Brad, what has gone wrong in this differentiation pathway for Wilms tumors? I mean, why is it blocked? I mean, you've shown us pretty convincing evidence. It looks good, but it's going nowhere. It's not getting mature. Where's the block?
Yeah, you know, Miguel Rivera has spent a lot of time very carefully looking at the regulators. It's blocked. He can tell you where it's blocked. It's blocked at a sort of an earlier than the field had thought, at a mesenchymal stem cell stage, where you can kind of look at the master regulators along the way, and you can kind of interpret exactly which progenitor it looks like. Why is it arrested there? There's data that well, certainly WTX and WT1 must be playing a critical role. There's data that Wnt signaling is involved in the arrest.
What do the mutations do to the differentiation pathway? Unanswered question.
I don't think that's known, but I think probably they affect Wnt signaling at some level.
Yeah.
So, because the majority of mutations are obviously occurring in intergenic or intronic regions as well, do you think the acquisition of an enhancer state, do you find that it correlates with either mutations in that region or microdeletions, or just from the sequencing data you've generated?
Great question. You're asking about it. So we've shown that the enhancers correspond to variants between individuals, somatic variants that are associated with things like lipid levels or immune disease. So now I think your question is, do you think some of the, we're looking so hard in cancer to document all the mutations in exons. Isn't it likely that there's a lot of mutations in the regulatory elements, and particularly those enhancers? And I couldn't agree more, but I don't have any data to show you yet.
But the evidence is fairly limited there, isn't it? I mean, in terms of your last slide, when you showed the regulation of gene expression, there are only a few of those things that are known to be mutated or translocated.
Well, so do you mean that there's very few of the transcription factors that are mutated, or
not the transcription factors, but the repressors, the regulators of epigenetics, the methylation or demethylation
only a few? Well, MLL2 is now turning out to be the most frequently mutated gene in all of cancer, isn't it?
That's one. EZH2 is two.
But I think actually the question here was are there sequence mutations in the regulatory elements, not in the regulators, but in the regulatory? Are enhancers a hotbed of getting mutated in cancer? Right. I think the answer is almost certainly yes, because it's another way of getting fitness. I'm not sure how it would help us strategize therapy. Yeah. But I would imagine yes.
It occurred to me, and maybe this is stupid, but it occurred to me that if you modify the repressive methylation or demethylation or the activating effects, you have very broad effects on the genome, and maybe those wouldn't be tolerated, so that the mutations in kinases are tolerated, but not something that affects such a broad pattern of gene expression. Does that make sense?
It makes total sense, but I guess you have to do the experiment to answer it. Scott would argue against it, I think.
We need more information.
So to address that issue a little bit is if you have, in C. elegans, for example, knockouts, they're viable. Yeah. You may see specific phenotypes in zebrafish. Yeah. We've done screening a variety of them, and we see that they're viable up to a certain point. Really? And there's examples in mouse models where the mice are viable as well. So.
And there's exquisite, you turn these regulators on and off during development, so that they probably have pretty potent effects on development. So
very specific.
I'd be optimistic.
Yeah, and I would argue that there are an ever-increasing number of mutated genes in cancer that are mutations in these histone-modifying enzymes, demethylases, DNMT3, very common in AML. Mm-hmm. So I think they're playing some role, but whether or not they're playing a role that induces a dependence, I think that will have to be worked out over time. And I think that's kind of what
That's what
getting
you're
at.
coming at. That's the perspective that Scott's sure you're coming at it from. I totally agree.